Hello, greetings, good morning, and welcome to the IMR-SEAMEO Webinar Series 2023. I am the moderator for today. My name is Melinda Dacre. The theme for today is Biomedical Research, the Cornerstone of Future Cancer Therapy. Institute for Medical Research, IMR, encompasses 8 centers. Today, the presenters are from Cancer Research Center. Cancer Research Center carries out research to improve prevention, early diagnosis, and treatment of cancers. Cancer Research Center consists of 3 units, namely hematology, molecular pathology, and stomatology. In the Malaysian scenario, cancer is the 4th leading cause of death. The 5 most common cancers are female breast, colorectal, lung, trachea and bronchus, lymphoma, and nasopharynx. Today's presentations will focus on lung cancer and nasopharynx cancer. Lung, trachea, and bronchus cancer is the 3rd most common cancer in Malaysia. It has the lowest 5-year relative survival at 11% and a median survival time of 6.8 months. Meanwhile, nasopharynx is the 5th most common site of cancer in Malaysia. It has a 5-year relative survival of 46% and a median survival time of 40.6 months. Today, the presenters and myself are wearing different shades of purple. We are also wearing this lavender ribbon. Lavender is a lighter shade of purple. Now, the lavender ribbon is a loop of ribbon worn in support of those affected by all kinds of cancer. The lavender ribbon is also worn to raise awareness about cancer in general. Before we begin the presentations, please allow me to make a few announcements about housekeeping. Questions will only be addressed after both presentations have ended. Viewers may post your questions using the question link provided in the YouTube channel. For those who would like to receive an e-certificate of participation, please fill in the separate link provided in the YouTube channel. And finally, please subscribe to our National Institutes of Health YouTube channel. Today we have 2 speakers. Please let me announce the first speaker. Let me introduce him to you. He is Dr. Sheikh Ahmad Kamal. Dr. Sheikh Ahmad Kamal is a research officer. He joined the Ministry of Health in 2011 and has been attached to the hematology unit since then. He has a PhD in cancer biology and oncology from University Putra, Malaysia. His research interests are stem cells and lung cancer. His projects include mesenchymal stem cells and gene therapy. He is also a production manager at a good manufacturing practice grade facility in IMR. He is an elected council member of the Malaysian Society of Stem Cell. Now, during his free time, he enjoys running, swimming, weightlifting, and music. Dr. Kamal will be talking about his research on mesenchymal stem cells and lung cancer. Dr. Kamal, you may begin your presentation.
Thank you, Dr. Melinda, moderator, for that kind introduction. I would also like to thank the organizing committee of the IMR-SEAMEO for having me here so that I can share some of my findings with everyone today. So good morning. My name is Dr. Kamal. I am from the Hematology Unit, Cancer Research Center, IMR. So today I'll be presenting a project entitled Trailing the Path of Genetic Engineering in Mesenchymal Stem Cell for Lung Cancer Treatment. As presented by Melinda, the incidence of lung cancer in Malaysia and globally is quite alarming. If you look into the cancer-related mortality, also lung cancer contribute as the major cancer-related death in Malaysia and also global. So because of that, there is a need for us to develop therapies, innovative therapies, so that we can enhance treatment efficacies for lung cancer patients. And hopefully by doing that, we can actually help lung cancer patients that have underwent chemotherapy to have a better life. So even though we have so many treatments modalities available for lung cancer patients, for example, chemotherapy, radiation, targeted therapy, none of this therapy is actually effective to eradicate the tumor. So 30 to 55% of lung cancer patients will actually develop relapse and die of their disease. So relapse is actually a condition where the tumor comes back after the treatment. So relapse or recurrence in lung cancer patient is actually due to a condition known as chemoresistant, where the tumor itself becomes insensitive to the treatment. There can be several reasons for tumor chemoresistance, and one of these reasons scientists believe is because of a very small population of cells known as cancer stem cells or tumor initiating cells. So cancer stem cells or tumor initiating cells you cannot destroy using common chemotherapy. However, you can identify cancer stem cells or tumor initiating cells using markers, for example, CD133, 44, 166A LDH. And when you stain these cells and you run flow cytometry, you will see that the population is very small, which is about 2.6% here, or 3.2%, which is lower than 5%. And if you sort this population out, and you run tumorigenic characteristic in an animal model, you will see that the tumor itself is actually bigger compared to the non-CSC, where these reflect the tumorigenic characteristic of the cancer stem cells. Trail, which is the name I use for my presentation, trail or trailing, is also known as tumor necrosis factor related apoptosis inducing ligand. So trail is basically a protein or a ligand. So upon binding of trail to its receptor known as DR4 and DR5, trail induces the intrinsic and extrinsic apoptotic pathways and subsequently leads to cell death. So trail itself has been shown very effective to destroy cancers and also cancer stem cells. And one of the good things about trail is that trail has a very strong tumor specificity, meaning that trail kills cancer cells or cancer stem cells without harming normal cells. However, there is certain limitations of using trail for clinical trial. Part of the reason is because trail has a very small molecular structure, which is about 30 kilodalton. So because of that, after infusion of trail to the patient, trail will easily get degraded by the body or excreted through the kidneys or the liver. And to overcome that limitation of using trail needed the delivery system. So you can use several ways to deliver trail. For example, you can use carbon nanotubes or liposome. However, I have chosen to use stem cells known as mesenchymal stem cells. So you can actually engineer mesenchymal stem cells to express trail and use these cells as a vehicle to deliver trail. So what is this mesenchymal stem cell? So mesenchymal stem cell is actually an adult stem cells that you can derive from several sources. For example, from bone marrow, adipose tissue, peripheral blood, dental pulp. One of the good things about mesenchymal stem cell that has favored mesenchymal stem cell for clinical transplantation is because mesenchymal stem cell has a very low immunogenicity. This is because MSCs has a very low expression of MHC class 1 and 2. And this also means that you can actually isolate mesenchymal stem cells from different sources and biobank and later transplant for inpatient for allogeneic transplant without the problem of having graft versus host rejection. MSCs also have a very strong proliferation rate where you can get up to 50 population doubling in 10 weeks. This also means that it is easy for you to expand mesenchymal stem cell up to billion cells and use it later for clinical transplantation, because for clinical transplantation you need abundant number of cells to be used in patients. And another characteristic of mesenchymal stem cell which has made MSCs an important cells or favor MSCs for cancer research is because of the ability of MSCs to actually migrate and home to the tumor environment. So this characteristic has been shown by several reports. For example, in this figure, you can see upon infusion of MSCs through the tail vein, MSCs is able to migrate and home to the tumor area, where in this picture shows a subcutaneous tumor model. subcutaneous tumor. And you can also see that MSC is also deposited highly in the tumor area here as compared to other organ. So because of the ability of mesenchymal stem cell to migrate and home to the tumor area, MSCs has been engineered to express anti-tumor agents, for example, cytokines, tumor necrosis factor or interleukins, and being used as a vehicle and studied in many cancer models for its efficacy. However, the limitations is that there is not much studies that has been made by scientists looking into the efficacy of engineered MSCs to target or destroy cancer stem cells, especially cancer stem cells in lung cancer. So for that, the objective of the study is that can we engineer mesenchymal stem cell to express TRAIL? And if you can engineer mesenchymal stem cell to express TRAIL, is these cells effective to destroy cancer stem cell, especially in lung cancer? And if MSC TRAIL can destroy cancer stem cell, what are the modes of action? Is it apoptosis or is it intrinsic? And last part is to see, can we actually use mesenchymal stem cell in combination with other chemotherapy, which is chemotherapy commonly used for the treatment of lung cancer. So the presentation can be divided into 3 parts. The first part is actually to produce the mesenchymal stem cell. The second is actually to isolate cancer stem cell and to perform verification and characterization. And the third part is actually to look into the efficacy of MSC expressing TRAIL, MSC TRAIL, to destroy cancer stem cell in a co-culture environment. So we look into the first part, if it's to produce the mesenchymal stem cell expressing TRAIL or MSC TRAIL. So to produce MSC TRAIL, I have chosen lentiviral transduction. So lentiviral transduction is basically one of the most effective ways that research have shown able to deliver gene to stem cell, especially mesenchymal stem cell. So to produce lentivirus encoding for TRAIL, first you need to transfect your host cells with the plasmid encoded for the lentivirus and also the plasmid encoded for the TRAIL. So for the TRAIL itself, I have tagged the TRAIL with a protein known as mCherry, which is also a red fluorescent protein, so that you can visualize your cells after transduction to see the effectiveness of the transduction process. So after you transfect your host cells, you get the lentivirus here, you concentrate the lentivirus, and then you transduce your mesenchymal stem cell, or another word, you infect your mesenchymal stem cell. So after infection, you will get a mixed population of mesenchymal stem cell expressing TRAIL and also wild-type mesenchymal stem cell. And after that, you can perform analysis to look into the transduction efficiency using flow cytometry, or to check the ELISA, to check TRAIL expression using ELISA, or mRNA using quantitative PCR. So this is actually a figure of mesenchymal stem cell which I have infected with the TRAIL encoding lentivirus. Since TRAIL itself is tagged with the red fluorescent protein, so you can see the MSCs is actually fluorescence red. And when you run flow cytometry, I managed to get about 80% of MSC TRAIL, which is very high, meaning that there is about 80% of the populations after transduction expressing TRAIL. And when I look into ELISA, I can see that the TRAIL expression from the MSC, transduced MSC, is significantly high compared to the control here and also the mRNA here. So based on this analysis, you can say that I have successfully produced mesenchymal stem cell expressing TRAIL. The second is actually to isolate cancer stem cells and also to perform characterization, which is the second part. So I have chosen 3 types of lung cancer known as A549, which is as a model for adenocarcinoma, H2170 as a model of squamous cell carcinoma, and H460 as a model of large cell carcinoma. And I have chosen CD133 positive as a marker for cancer stem cell. So CD133 positive has been shown one of the most effective markers you can use to isolate cancer stem cells in different tumor models. So if you look at this, dot plot, the cancer stem cell is about 0.1% in all of 3 lung cancer cell lines. So I sorted out this cancer stem cell population and I performed characterization. So characterization is based on sphere formation and also clonogenicity. So based on the sphere formation assay, you can see that the positive population cancer stem cell exhibit greater sphere size compared to the non-CSC here. So this also shows that the cancer stem cell is able to grow in an encouraged independent medium, which is one of the most important characteristics of cancer stem cell. The second here, you can also that the cancer stem cell has a higher clonogenicity compared to the non-CSC or non-cancer stem cell. So this also shows that this cancer stem cell can be grown in a very low number of cells, where it does not need neighboring cells to help them to grow. So these two assays is actually one of the most important assays you have to perform to characterize a cancer stem cell. And the last part is to perform assays looking into ALDH activity. So ALDH is actually an aldehyde dehydrogenase enzyme, where this enzyme is used by cancer stem cells to pump out treatment from the cells. So you would expect cancer stem cells to have a very strong expression of ALDH. And you can perform flow cytometry looking at ALDH expression. So if the cells are positive, you will get the positive population here compared to the negative here. So based on the analysis, you can see that the cancer stem cells, the CD133 positive cancer stem cells, exhibit or express very strong ALDH activity as compared to the non-cancer stem cell in all three lung cancer cell lines here. So the last part is that to look into ability of mesenchymal stem cell to actually target cancer stem cell in a co-culture environment. So theoretically, you will have lung cancer cells or lung cancer stem cell expressing the TRAIL receptor known as DR4, co-culture with mesenchymal stem cell expressing TRAIL here. So in a co-culture here, upon ligand and receptor mediated binding, that would actually induce apoptosis or cell death to the tumor and cancer stem cell. So this is actually a video showing a time-lapse images of co-culture between MSC TRAIL and also lung cancer stem cell. So I've seeded first the lung cancer cells or lung cancer stem cell at the bottom of the flask here, and the next day I added MSC TRAIL, which is all these rounded cells here. So from here you can see from the video how actively MSC TRAIL is actually moving and targeting and destroy the cancer cells or cancer stem cell. So this culture was performed for 48 hours of co-culture. So after several hours, days, and even 2 days and 4 days, you can see how actively MSC TRAIL is killing the cancer stem cells here. And at the end of the experiment, you can see that there is a lot of debris and dead cells which comes from the lung cancer cells here. And at the end of the experiment, you can have almost 90% of the tumor actually died from the treatment itself. So I performed a viability assay to see the ability of MSC TRAIL to inhibit lung cancer stem cell proliferation, and result shows that the MSC TRAIL was able to inhibit the proliferation of cancer stem cells here in 2 of these lung cancer cell lines, which is H4C0 and H2170 here. Apoptosis assay is another assay that I performed. So the principle is that for a normal cells, the expression of phosphatidylserine is usually in the inner part of the cellular membrane. However, if the cells is undergoing apoptosis, the phosphatidylserine will be flipped to express out at the outer part of the cellular membrane. So this phosphatidylserine can be detected using adenosine-5 and tagged with a fluorescence. So using a fluorescence here, you can actually run flow cytometry and quantitate the percentage of cell expressing the phosphatidylserine or adenosine-5. And from the analysis, you can see that after treatment using MSC TRAIL, MSC TRAIL was able to significantly induce apoptosis to the cancer stem cell population here as compared to the control. And you can also see that the total effect of MSC TRAIL here is even higher compared to the recombinant TRAIL here. Recombinant TRAIL is actually a drug where has been used in clinical trial. So based on this analysis, you can see that the effect of MSC TRAIL is even greater compared to the treatment, compared to the drug used in clinical trial. Dead cells is another assay that I perform. So if the cells are dead cells, you will get green fluorescent protein or something like this, and you can perform flow cytometry. If the cells is already dead cells, so you will get green fluorescent cells like this. So when you run flow cytometry, this is the population here. So based on the analysis, you can see that MSC TRAIL was able to significantly kill the cancer stem cell here as compared to the control, and this can be seen in 2 of the lung cancer cell lines, which is H4C0 and H2170. So from here you can see that for A509, you can see that A509 is actually resistant to the effect of MSC TRAIL because there is no different in the viability here compared to the control. So I sensitized A509 using a common chemotherapy used in lung cancer patient, which is cisplatin and 5-FU. And upon sensitization, you can see that sensitization using common chemotherapy can indeed enhance the effect of MSC trail here as compared to the control. So these results also can also means that for lung cancer patients that have underwent first-line chemotherapy, that chemotherapy can actually act as a chemosensitizer to enhance the effect of MSC trail. So if you would like to know more about my study, you can have a look at some of the publications that I have reported in several journals. Some of the reports that I have published in several journals. So the future studies, some of the future studies that we can actually embark is actually to look into an in vivo, because based on my analysis, I perform in an in vitro data. So we can actually perform in an in vivo analysis to look into a bigger animal model or a bigger sample size to look into efficacy by distribution and fate. And exosome is also another area, exciting areas that for those who are doing MSC's research. So scientists now believe that exosome, not just mesenchymal stem cells, but exosome also can actually induce a therapeutic effect, the same as MSCs. So looking into exosome derived from MSC trail is another area which I believe is quite interesting that scientists can embark in future. And the last part for future studies that we can also look into what are the genes involved. For example, genes that are related to trail resistance, and by targeting these genes, you can actually use that kind of method to enhance the effect of MSC trail. So that's about it. I would like to say thank you to people from my center, the Cancer Research Center. And as this project is also part of my PhD project, I would also like to thank my supervisor from University Putra Malaysia, Dr. Sharil, and also my co-supervisor, Shariza and Prof. Rosita. So thank you very much, everyone. So that's about it, my presentation. If you have any questions, you can directly email to me through the email here. I'm also very open to accept any collaboration with anyone interested for the project. Thank you very much. So I pass this screen to our moderator.
Thank you. Thank you, Dr. Kamal, for that fascinating sharing. To our viewers, if you have any questions, kindly post them in the question link provided in the YouTube channel. Next, let me introduce to you our second speaker, Madam Marini Binti Marzuki. Madam Marini is a research officer. She joined the Ministry of Health in 2015 and has been attached to the Molecular Pathology Unit since then. She has a Master of Science in Human Genetics from University Science Malaysia. Her research interests are in immunotherapy and cancer biology. Her projects include circulating tumor cells for the prognosis of breast cancer and drug discovery involving natural and synthetic compounds. She is the Secretary of the Malaysian Society for Human Genetics. She is also a lifetime member of the Nasopharyngeal Carcinoma Society of Malaysia and an ordinary member of Persatuan Genetik Malaysia. During her free time, she enjoys arts and crafts, decorating, designing, movies, and music. Madam Marini, please share your presentation with us.
Assalamualaikum and a very good morning to all. Thank you, Dr. Melinda, for the kind introduction. I am Marini, Research Officer from Molecular Pathology Unit, Institute for Medical Research, IMR. I am grateful to have all of you this morning for the 2023 IMR-SEAMEO webinar series. Today, I will share our experience on patient-derived xenograft, an invaluable resource for the development of tools to fight cancer. I will first give an overview of cancer, its incidence and challenges. Then, I'll briefly talk on the development of patient-derived xenograft PDX model, the PDX model applications, and finally, summary and future perspective. So, what is cancer? What are the most common cancer in Malaysia? How about the respective survival rates? Cancer can start almost anywhere in the human body, which is made up of trillions of cells. Normally, human cells grow and multiply to form new cells through a process called cell division. When cells grow old or become damaged, they die, and new cell will take their place. Sometimes this orderly process breaks down. Abnormal or damaged cells will grow and multiply to form abnormal mass or lump of the cells in our body. Cancer is a disease whereby our body cells grow uncontrollably and spread to distant organ or lymph node. Overall, there were nearly 49,000 new cases recorded in Malaysia last year, and this number is expected to double by 2040. The rising number of cancer cases will become a major health issue as they continue to put physical, emotional, and especially financial strain on the patient, the immediate family, communities, and the country healthcare system. The most common cancer in Malaysia are breast cancer, followed by colorectal cancer, lung cancer, nasopharyngeal cancer, and liver cancer. Breast, colorectal, and lung cancers make up about half of the total cancer cases in our country. As mentioned, female breast cancer is the most common cancer in Malaysia, of which there were more than 20,000 cases within 5 years. However, it shows a high survival rate of nearly 70% compared to other cancer. On the other hand, although nasopharyngeal cancer, NPC, is the 5th most common cancer among Malaysians, these patients recorded less than 50% of the survival rates within 5 years. Also, Southeast Asians have the world's highest age-standardized incidence and mortality rates of NPC. Hence, there is an urgent need to improve patient survival rates by finding new treatment therapies or improving the current treatment regimes in hospitals. In the laboratory, such research intention is often hampered by challenges. For example, in NPC research, the patient cells are difficult to be cultured. There is no commercial NPC cell line currently available in the market. Most shared NPC cell lines were found to be somatic cell hybrids with HeLa cells, a type of cervical cancer cells. Hence, many published work using NPC cell lines in the world are later found not to be conducted on authentic NPC cells. Additionally, these NPC cells models are not from Southeast Asian patients. The following slide will be on our experience on developing patient-derived xenografts to study cancer in IMR as a research solution to the previously mentioned lab challenges. But first, what is patient-derived xenograft, or PDX in short? Well, PDX is a tumor xenograft which is derived from the transplantation of fresh patient tumor specimen into immune-compromised or immunodeficient mice. It allows the patient tumor specimen to be expanded or propagated in the mice for numerous generations. PDX can also maintain the molecular, genetic, and histological heterogeneities that are typical of tumor of origin. With this, PDX is a good tool to study cancer biology and therapeutics. In IMR, we have developed PDX model for NPC research. Cancer tissue from the patient's nasopharynx will be processed in the lab using enzymatic digestion to become single-cell suspension, semi-digested, or tissue fragment. The resulting cells or fragments will be grafted into an immunodeficient mouse. Engraftment can be performed in the capsule of mouse kidney, under the skin or subcutaneously, and at the mouse nasopharynx orthotopically. As I mentioned earlier, PDX can be established through these 3 common engrafting methods. There are several mouse strains that are commonly used for xenografting, which are nude mouse, not SCID gamma, NSG, and severe combined immunodeficiency mouse, SCID mouse. In our lab, we are mostly use NSG and SCID mouse strains. The successful xenograft can be cryopreserved in liquid nitrogen, continuously maintained as xenograft in the mice, or cultured in vitro as monolayer cells or spheroid. This slide shows one example of our successful PDX establishment from a 73-year-old Bidayo female patient who was diagnosed with undifferentiated type 3 MPC in 2016. After taking the biopsy specimen for diagnostic tests in the hospital, some of their remaining biopsy tissues were implanted into renal capsule of a NSG mouse in our lab. Upon successful first-generation establishment of the PDX, various characterization stainings had to be performed before it can be used as a reliable system to study MPC in the lab. The PDX histology resembles the patient MPC histology, which is non-keratinizing and differentiated subtype. It is an epithelial growth that is also negative for leukocyte as shown here. The PDX is positive for Epstein-Barr virus, EBV. This is very important as MPC is almost always associated with the presence of this particular virus. After successfully developing and characterizing the PDX model, we went a step further by establishing PDX that can also express green fluorescent and luciferase 2 protein, or in short GFP-LUC2. To establish this model, we first transfected plasmid that carry out the required gene to produce lentiviral expressing GFP-LUC2 into a producer cell line called HEC293 T cells. Once the replication inactive lentiviral particles were produced by HEK cells, they were harvested and transduced into the PDX cells. The mouse with the resulting growth was viewed under an in vivo imaging system to verify the presence of GFP-positive cells. The GFP expressions in the excised growth can also be viewed similarly. There are several applications of the PDX model. In IMR, we have used the PDX model in cancer metastasis, drug therapeutic, as well as immune-related studies. For the establishment of the metastatic model, we inoculate the GFP-LUC2 PDX cells into the mouse by intracardiac route. After the injection of the cells, their distribution was observed via luminescence imaging to ensure the cells were distributed well in the mouse after inoculation. Bioluminescence signal were then captured every week to monitor for any tumor growth at various sites in the mouse. The mouse will be euthanized after the experiment endpoint was reached. Usually, the endpoint will be the tumor growth reaching a certain volume as permitted by the Animal Ethics Committee. 3D image of GFP-LUC2 PDX can be viewed using IV spectrum in vivo imaging system. This system helps us to identify precisely the source and the depth of the tumor, as well as to find other sites of tumor spreads, as viewed in this video. The IV spectrum in vivo imaging system can facilitate non-invasive longitudinal monitoring of disease progression, cell trafficking, and gene expression patterns in living animals. Animal use in research must be guided by the 3R principle, which are replace, reduce, and refine. These principles were developed to provide a framework for performing more humane animal research. For more info on the 3Rs, you may go to this website. In order to fulfill the 3R principles to reduce animal usage, we embark to culture the PDX cells in vitro. The following slide shows our works using PDX cells that were successfully maintained as short-term culture. For evaluating drug therapeutic efficacy, we compare the PDX cells grown in the form of 2D monolayer and 3D spheroids. The tumor was processed to get single cells. Later, these cells were cultured in vitro. 2D and 3D cells were treated using drug A, which is a standard chemo drug for NPC, together with approved drug for other diseases which are called drug B and drug C. To evaluate cellular response following drug treatment, viability was assessed using cell titer glow luminescent cell viability assay. The data were then used to construct this dose response curve. Our result shows that 3D spheroid require a higher dose of drugs to kill the cancer cells compared to the 2D model. This image shows NPC tissue section stained with H&E. As you can see, NPC tumor clusters are infiltrated with immune cells. Yellow dashed line here is the example of an NPC tumor cluster, while the red arrow indicates the presence of immune cells that included natural killer cells. So natural killer cells are the preeminent cytotoxic innate lymphocytes which have been originally described for their spontaneous cytotoxicity against infected and tumor targets. NK-based cell-based therapies have been shown to be increasingly promising in the therapies of cancer, thus affirming the role of NK cell in the eliminating of cancer cells. The established PDX will serve as a panel of study models and be adapted for in vitro co-culture assays as shown in the next slide. For the co-culture assay, we use a NK cells line NK92 to evaluate its killing effect on the cultured PDX cells. Viability was assessed using two techniques, which is cell titer glow and a real-time cell analysis method via the xCelligence system. This slide shows the result of a co-culture experiment between NK92 and cancer cells using the xCelligence system. With the addition of NK92, the NPC cells were gradually killed over time accordingly to the ratio of NK92 added. Figure 11 looks at the 50% killing time by NK92 when co-cultured with NPC cell line or PDX line. With a higher ratio of NK92 co-cultured with NPC, the cell killing process were more efficient, thus leading to a shorter 50% killing time. So in order to visualize NK92 killing the target NPC cells and the effect of its killing in the NPC cells at various target and effector ratio, a parallel experiment of NPC transduced with GFP-LUC2 were co-cultured with NK92. So in this experiment, we use NPC cell line as the target and NK92 as the effector. Ratio 0 to 1 is a negative control, which means no target cells, only effector NK92 was seeded, while as a positive control, we seeded only target NPC cells at the ratio of 1 to 0. Cell morphology changes were monitored by acquisition of fluorescence image at 24 and 72 hours. GFP positive image denote viable NPC cells. Majority of target NPC cells were still viable at a lower TE ratios. Red images are non-viable NPC cells, especially seen at a higher TE ratio. Figure 12 provide the visual evidence that corroborated with Figure 11's data that NPC cells were killed at a TE ratio dependent manner by NK92. Before I end my sharing session, I would like you to remember these key points. Patient-derived xenografts, PDX, are extremely valuable for the development of tools to fight cancer. In IMR, PDX are utilized in the development of cancer metastatic models, preclinical drug testing, and studies for evaluation of immune cell cytotoxicity. More resource and effort are required for the establishment of models from local patients of different cancer types. I would like to take these opportunities to thank all the patients and clinicians who are willing to contribute to biomedical research, our past and present staff of Molecular Pathology Unit, Cancer Research Center IMR for their hard work and dedication in cancer research, and finally, our Head of Cancer Research Center, Director of IMR, and the Ministry of Health for their support in cancer research. And with that, thank you very much for your attention. If you have any query, feel free to contact me through my email. I'll try my best to address them well. Now, I pass the floor back to our moderator, Dr. Melinda. Thank you.
Thank you, Madam Marini, for that visually appealing and compelling presentation. Now we shall move on to the question and answer session. To our viewers, if you have any questions, kindly post them in the question link provided in the YouTube channel. Please give me a moment to scan through the questions collected. Please keep your questions coming. Wow, it seems like we have received quite a number of questions. Question 1 goes out to Dr. Kamal. Dr. Kamal, why is mesenchymal stem cell preferred over other stem cells? This question comes from Ed.
Thank you, Ed, for that question. So basically, the difference between mesenchymal stem cell and other kind of stem cell is actually the inherent capacity of MSCs to migrate and home to the tumor environment. So that is actually one of the most important characteristics of MSC that favor these cells for cell-based therapy, especially in cancer. So compared to other stem cells, you can actually engineer these cells to express any ligand. However, does this cells or stem cell is capable to bring the ligand or the cytotoxic drug or anything directly to the target area? So that is the question, and that is something that can be done by MSC. So that's the reason why I have chosen mesenchymal stem cell as the preferred choice of cells for cell-based therapy as compared to other kind of stem cell. Thank you.
All right, thank you, Dr. Kamal. The questions are coming in. Question number 2. Question number 2 is from Brad. Brad would like to ask Madam Marini. Madam Marini, what is the significance of having a patient-derived xenograft originating from the tumor of a patient of Bidayuh ethnicity?
Okay, thank you, Brad, for your question. Up till now, standard care of MPC has been on research outcome from China and Hong Kong. However, cancer is a group of heterogeneous disease. So, different genetic background may lead to different risk, pathogenesis, and respond to the treatment. For example, in Asian patient, in Asian female actually, specifically Asian female non-small cell lung cancer patient have the higher frequency of EGFR mutation compared to the Caucasian. Due to this reason, Asian female patients are more responsive to tyrosine kinase inhibitor as the first-line treatment of this cancer. So therefore, it is crucial for us to generate cancer models from local patients to develop these treatment regimes that are more suitable and effective for them. So I hope I have answered your question.
Thank you, Madam Marini. Keep the questions coming. Please give me a moment to scan through the questions. Right. The questions are coming in. Madam Marini, you have the third question. Madam Marini, what are the advantages of patient-derived xenograft compared to other cancer cell lines? And this is a question from Aliyah.
Okay, Aliyah, I appreciate your asking. So this is what I think. The most important advantage of PDX over cell line is that they are histologically similar to the original patient. So, tumor heterogeneity is also retained from the original tumor in PDX. Whereas in cell line, the similarities are lost through time because the serial passaging during in vitro cell culture. So, moreover, cell lines are established and grown in artificial environment, such as using plastic culture vessel and culture media. For this reason, PDX offer greater clinical and biological relevance than traditional cell line grown as monolayer. So, thank you.
Right. Thank you, Madam Marini. We move on. We have a fourth question now. And question number 4 goes out to Dr. Kamal. Dr. Kamal, your question is from Mariah. Mariah asks, what are the differences between MSC trail and CAR-T? And what makes MSC trail superior to CAR-T in clinics?
So thank you, Mariah, for that very good questions. So CAR-T is actually an engineered immune cells, engineered T cells that you engineer these cells to express a certain antigen, a certain CAR, for example, CD19 and 20. So the limitation of using CAR-T is that the antigen recognition, which is CD19 and 20, is highly expressed in hematological malignancies but not in solid tumors. So there is no doubt CAR-T is always the best treatment for hematological malignancies. For example, in diffuse large B-cell leukemia or some multiple myeloma. But it is not actually a very good treatment for solid tumors. Part of the reason is because the antigen recognitions and also the condition of the tumor itself, which are mostly does not favor T cells to go into it. So MSCs expressing trail, on the other hand, is cells which has been shown by reports to have an inherent capacity to migrate and home to the tumor itself. And also the receptor that trail target, which is DR4 and DR5, has been shown to be highly expressed in solid tumors, especially lung cancers, compared to normal cells. So you have a very specific targeting kind of ligand for that kind of therapy using MSC trail. So I hope that answered that question. Thank you very much.
Thank you, Dr. Kamal, for your answer. Let me scan through again. Dr. Kamal, you have another question. This is from Zawana Hansari, right? A question from Zawana Hansari. Can lentivirus transfection be utilized to express microbial protein in mammalian cells? Dr. Kamal?
Thank you, Zawana Hansari, for that very interesting questions. So, microbial protein in mammalian cells, I think it should be okay because lentivirus is a virus that infects mammalian cells. It's just that you need to know the sequence of the protein that you will want to express, and from that sequence, you like it with a plasmid encoding with the lentivirus itself, and then you can use that lentivirus to infect mammalian cells. I think it should be okay, no problem. Thank you.
Thank you. Right. Right, let me see. We have a question from HUSM from Lee Shen Han. Lee Shen Han of HUSM. Question to Madam Marini. How are the patient-derived xenograft cells implanted orthotopically into the nasopharynx of mice? And is this technically challenging? Madam Marini?
Thank you, I think Mr. Lee Shenhan from USM for the question. Basically, PDX cells will be implanted orthotopically at the nasopharynx base at the mouth, which is here, and then it is a bit challenging compared to the intravenous route. So yes, it is technically challenging, and you need certain technical expertise to do that, because sometimes the cells will move to other part or maybe leaking. So yes, you need technical training for that particular route to be implanted in the mouse. Thank you. I hope I answer your question.
Thank you, Madam Marini. Thank you. We still have time for another question. This one is from Muhammad Fazli Shaharuddin. Muhammad Fazli Shaharuddin of HKL. And the question is for Dr. Kamal. Dr. Kamal, is there any potential risk of mesenchymal stem cell therapy, such as the probability of inducing pro-tumorigenic differentiation into undesirable tissue, or any other serious side effects? Dr. Kamal?
So thank you, Fazli, for that interesting questions. When you say about side effects, a side effect you can say in the form of graft versus host rejection. So I wouldn't say MSc would have a problem when it comes to side effects of GVHD, because MSCs, as you can see, it has a very low expression of MSc class 1, MSc class 2. So side effect based on GVHD will not be a problem. And another question is that can MSc induce tumorigenic characteristic or tumor to the host? So I've read several papers looking into the long-term effect of MSc transplantation. So I can say that these studies have shown that there is no evidence that MSc can induce formation of tumor. So it should be safe. So basically, I believe that using MSc for transplantation in patients, I believe is safe, is quite safe. However, you may need to prove that concept in a preclinical model, because proving that concept in a form of efficacy, safety, and toxicity is important for you to have a data that you can convince regulators later when they use that for clinical transplantation. So you need to prove that, because sometimes MSCs coming from different sources would have different effects. So you need to prove based on efficacy, toxicity, and other data on your preclinical model first. Then you can convince the regulators. And another question is that can MSCs maybe differentiate into other kind of cells? So differentiation of MSCs is limited into 3 only: adipocyte, osteocyte, and chondrocyte. And differentiation of MSCs into this lineage is not something simple, because you need to have a very specific cocktails of medium. Then you can induce these cells to differentiation. So I don't think that there will be a problem for MSCs differentiate into other kind of cells once MSC is transplantation, because differentiation itself is a very hard process. You need to have a specific cocktail and a specific condition. So hopefully hopefully that answered that question. Thank you very much.
Thank you, Dr. Kamal, for the comprehensive answer. I think we still have time for another question. This one comes from Lee. Lee, your question is to Madam Marini. Lee asks, why is it important to work on nasopharyngeal carcinoma, given that other cancers have higher incidence rate and poorer survival?
Thank you, Mr. Lee, for your question. And yes, you are right in highlighting this. In Malaysia, one of the cancer with a low 5-year relative survival rate is lung cancer. So its statistics are also pretty low in the West. For example, in the US, there's around 10 to 30% of 5-year relative survival rates of all stages. In view of this, a lot of resources from the West have been poured into lung cancer to improve patient survival and quality of life. In fact, the most advanced targeted therapy currently available and developed by the West for lung cancer. So as MPC is only largely found in Southeast Asia and the East, it has not received as much attention as lung, liver, or stomach cancer, all with lower survival rates than MPC. So from the research and pharma community in the West. So from there, that's why we need to do more research on MPC to help our local patients to try to improve their treatment. So I hope I answer your question. Thank you.
Thank you, Madam Marini. And the questions are coming in very fast. So we still have time. This is a question from Siti Hajar Shahid. Siti Hajar Shahid asks, Dr. Kamal. Dr. Kamal, do you see any potential of the MSc trial combined chemotherapy to be further studied for clinical efficacy?
So thank you, Siti Hajar, for say based on my understanding and also the experiment or studies that I have performed. So I would say yes, MSc trail has a very strong potential to be used with other chemotherapy, because studies have shown that certain chemotherapies, for example 5-fluorouracil or cisplatin, can actually enhance the effect of trail itself, because there is certain condition where trail may not be in an effective treatment towards trail-resistant cells. So you can actually sensitize these cells using common chemotherapy as a first-line treatment to kill the tumor first, and then after killing the tumor using the chemotherapy, that can act as a chemosensitizer to enhance the effect of trail itself, where trail or MSc trail is used later after that treatment. So I believe by combining both will be a very good area, or a very good ways for you to develop treatment for lung cancer patient. Not just lung cancer patient, maybe for other type of cancers also can. Thank you.
Dr. Kamal, your mesenchymal stem cell topic is very hot. You have another question, also on mesenchymal stem cell, from Farhana. Farhana asks, has mesenchymal stem cell been used in Malaysia transplantation clinical setting?
When it comes to transplantation in Malaysia, you need to go through regulators, for example, NPRA, National Pharmaceutical Regulatory. So when you say, is there any transplantation in clinical setting, I believe that under MOH, Ministry of Health, there is no transplantation yet, which from us or from MOH. But however, from clinics or from private institutions, I'm not sure. Maybe they have. Maybe they have, and maybe they have go through proper channels. I believe that when you ask, under MOH, there is none. But however, under other institutions, maybe there is. So hopefully that answered the question.
Thank you, Dr. Kamal. Yet again, another question for you, and again on mesenchymal stem cell. This time, this question is from Ridwan. Ridwan asks, how is the mesenchymal stem cell trail specifically derived? What are the possible challenges of mesenchymal stem cell clinical trial?
When you say, thank you, Dr. Ridwan, for that question. So, how are MSc trails specifically derived? So, I did not derive directly another word that get a pure population of MSc trail, because to do that you need to perform sorting or isolation. So I didn't do that. However, if you want to derive specific MSc trail, you can actually use sorting, for example, using ARIA to isolate pure population of MSc trail, so that you will not have mixed population of other MSCs, for example, wild-type MSCs. And another question, your question is that what are the challenges of MSc in clinical trial? So I believe the challenges is actually to prove that concept of efficacy, toxicity, and non-targeting effect or things like that. Because you need to convince the regulator that your cells, the cells that you produce under your institute or under your university or anyway, is actually safe and also highly efficacious to treat a certain disease. So that is, for me, that is the challenge that you have to face. And also for cells to be used clinical transplantation, especially in Malaysia, you need to produce in a GMP certified lab. That is another challenge, because not everyone in Malaysia has a GMP certified lab. However, that is a crucial requirement. So that will be another challenges for MSc to be used in clinical trial. Thank you very much.
Thank you, Dr. Kamal. I think you picked a very good topic to discuss today. Right. Ladies and gentlemen, I'm afraid that is all the time we have for addressing questions. We have come to the end of this webinar session. Now, in conjunction with the 123rd anniversary of the establishment of the Institute for Medical Research Malaysia, we will be having an IMR Open Day scheduled on the 19th of October 2023. There will be activity booths, exhibitions, exciting and fun competitions, all for free, and it is open to the public. So see you there. Until then, that is all. Thank you from all of us from the Institute for Medical Research Malaysia. Take care, stay safe, and bye-bye. you.